All right, good afternoon, everyone. As Kelly mentioned, I'm Kayla Minesinger. I'm a fourth-year Ph.D. student in our biomedical engineering program, and I work with Dr. Raj Guru in the neurotherapeutics laboratory on the first floor. And it's, to my knowledge, I think we're the only hearing lab in the building, so this might be a little bit different than good one. We're the only hearing lab in the building, so it might be a little different, so just bear with me. And as Kelly said, I'll be presenting some of my thesis work and a project I've been leading in our lab, which is focused on exploring the impact of noise exposure and cochlear implantation on hearing preservation outcomes. To begin, I wanted to do a brief review of the anatomy of the inner ear. Today, our organ of interest is the cochlea, which is the spiral-shaped organ divided into 3 fluid-filled chambers, the scala vestibuli, the scala media, and the scala tympani. The organ of Corti is the sensory organ located within the scala media of the cochlea and is responsible for transducing sound vibrations into neural signals. The basilar membrane and the tectorial membrane are 2 important structures that interact with the cochlea's hair cells, and they act as the sensory receptors of the cochlea. So there's 2 types of hair cells. The outer hair cells are OHC and inner hair cells, IHC. Inner hair cells convert sound vibrations into electrical signals sent to the brain, and then the outer hair cells amplify sound vibrations with frequency-specific sensitivity to sound. To dive a little deeper into the cochlear mechanics, it's really important to know that the frequency mapping of the cochlea allows us to perceive distinct pitches and complex auditory information. The cochlea's hair cells and neurons are sensitive to stimulus intensity and frequency, being most responsive at their characteristic frequency. In fact, this intricate hearing organ has a frequency mapping shown on the left, with high frequencies detected at the base and low frequencies at the apex. And understanding this tonotopic mapping is essential for my lab's research in order to identify region-specific damage, which can affect hearing restoration abilities via cochlear implants or even potential therapeutics. Next, I want to introduce noise-induced hearing loss. NHL is the 3rd most common chronic physical condition in the United States, affecting 24% of adults in one or both ears due to recreational or occupational unsafe noise exposure. And whether you realize it or not, in our daily routines, it's really easy to become accustomed to the noise you might experience. And it's well documented that excessive noise, whether we perceive it as loud or not, can lead to irreversible hearing impairment. And given this fact, NIHL is actually categorized as a type of sensorineural hearing loss, meaning it's a type of hearing loss that results from damage to the inner ear structures, specifically the hair cells I mentioned, or even the auditory nerve afferents themselves. To better understand how NIHL leads to hearing impairment, we need to really delve into the underlying biological processes. The pathophysiology of NIHL involves a complex interaction between mechanical, biochemical, and cellular damage within the auditory system. For example, mechanical stress from loud noises directly damages the hair cells, which are crucial for converting sound vibrations into neural signals. Since these cells don't regenerate in humans, their loss leads to permanent hearing impairment. Additionally, noise triggers oxidative stress, releasing reactive oxygen species that further damage cochlear cells. The release of neurotransmitters like glutamate overstimulates neurons, resulting in excitotoxicity, and this causes further cell death. And then finally, noise exposure can also impair the cochlea's blood supply, and this leads to ischemia and overall compounding the damage. So these combined factors lead to the gradual and inevitable irreversible hearing loss characteristic of NIHL. Because NIHL is the only form of preventable hearing loss, the best approach to target this remains taking preventative measures and staying educated and aware of the risk of noise exposure. Some other current treatments for managing the condition include pharmacological treatments and corticosteroids. However, with the absence of FDA-approved therapeutics for hearing loss and the nature of this irreversible damage, hearing aids are the most common treatment for hearing loss and are well studied, are well suited for those who have mild sensorineural hearing loss. Hearing aids work by amplifying sound, and it allows people with hearing loss to communicate a little bit better, but they amplify sound rather unselectively, so even in comfortable, familiar, noisy environments, it can be challenging for users to communicate effectively due to these limitations. So in all, mild NHL can be managed with hearing aids. However, in more severe cases, especially those involving widespread damage to hair cells and the neural pathways, cochlear implants may be the chosen treatment modality. As one of the most highly successful neuroprosthesis to date, CIs have benefited more than 1 million patients worldwide to treat sensorineural neural hearing loss. Cochlear implants are surgically implanted into the basal region of the cochlea to restore hearing for people with severe hearing loss by bypassing damaged parts of the cochlea. The implant captures sounds, converts it into electrical signals, and directly stimulates the auditory nerve, allowing the brain to interpret these signals as sound. Many cochlear implant recipients experience significant improvements in speech understanding, even in noisy environments, and report enhanced quality of life through better communication. Something I want to talk about a little bit in depth today is cochlear implant candidacy, which is determined based on several factors, including audiometric criteria. So candidacy is generally defined for individuals with moderate to profound sensorineural hearing loss who don't achieve significant benefit from hearing aids. Candidacy is determined using clinical audiograms like the one shown here on the left, which measures a patient's hearing thresholds across a frequency range. In this example, the thresholds fall within the moderate to profound hearing loss range, particularly at the higher frequencies. And this hearing loss profile highlights the importance of different frequency hearing regions, and I just want to break it down quickly. So if we start on the left side of the graph, in the lower frequency region, we see relatively near normal hearing thresholds, and these lower frequencies are critical for speech intonation, sound localization, and music perception. As we move into the mid-frequency range, which is essential for speech understanding, these hearing levels begin to decline, and that leads us to the high-frequency region, where you see that steep drop-off in the hearing levels. And this region is essential for speech consonants, enhancing sound detail perception, and understanding speech in the noisy environments. And this, again, is where the hearing loss becomes most pronounced, and this is typical of a CI candidate. This profile underscores two main points I want to make. One, CIs are imperative for addressing the high-frequency hearing loss, and two, patients more often than not have some sort of residual hearing, typically in the lower frequency regions, that must be preserved post-implantation. And then lastly, to relate this back to the noise-induced hearing loss, the extent of noise damage can often push patients into this candidacy range, kind of similar to this. And NHL can create these similar hearing loss patterns, particularly in those higher frequency regions where the damage to the hair cells and neural pathways is most pronounced. So this is why understanding how noise exposure affects the cochlea and neural structures is so important for intervention planning. So now, having said all of that, and given the fact that cochlear implants are in fact highly effective, preserving hearing and minimizing surgical trauma remain a critical challenge in our field. In recent decades, CIs have undergone significant technological advancements aimed at preserving residual hearing, particularly in the low frequencies, which again is crucial for speech perception and overall auditory experience. Early strategies, such as the use of short electrode arrays and soft surgery techniques, have shown promising outcomes, with a substantial percentage of patients actually being able to maintain those lower frequency hearings. However, the outcomes can tend to vary, with as little as 30% of patients or more experiencing some sort of degree of delayed acoustic hearing post-implantation. This has been thoroughly documented through a series of retrospective chart reviews and a few clinical trials, in which research indicates that factors such as the duration of deafness, age of implantation, etiology, and surgical techniques influence these outcomes. Noise-induced hearing loss, in particular, has been associated or suggested that that there is more of a severe post-implantation hearing loss due to the exacerbation of existing cochlear damage due to the electrical stimulation. A couple animal studies have also demonstrated some loss of viable neural elements resulting from mechanical insertion trauma, as well as long-term electrical stimulation. So one significant reason for these complications following CI is electrode insertion trauma. Electrode insertion trauma, or EIT, refers to the mechanical damage caused to the delicate structures of the cochlea during the surgery due to the electrode array. This trauma can manifest in various ways with multiple biochemical mechanisms occurring as a result, affecting specific cochlear structures and the neural substrates. So for example, oxidative stress causes cellular damage leading to cell death pathways. Altered blood supply and ionic homeostasis causes malfunction of appropriate nutrient delivery to cochlear cells and ultimately disrupts proper functioning and signaling in the cochlea. So while preserving hearing is the primary goal, achieving this can be complicated because of EIT and is a key factor that can further compromise the cochlear function during implantation. Okay, so thus far we have reviewed NIHL and EIT, and clearly these two types of traumas are very well studied. However, these two traumas have only really been studied independently, and there's a lack of consensus in our field regarding their independent contributions or potential detrimental interactions in hearing preservation outcomes. And further, there's a paucity of data really characterizing the underlying pathophysiology responsible for these undesired outcomes. So essentially, my lab work has aimed to fill the critical gaps in our understanding of the interplay between NIHL and EIT, and then their combined impact on cochlear implant outcomes. So by advancing this knowledge in these areas, the study has the potential to inform the development of more effective therapeutic strategies, ultimately improving the quality of life for individuals with hearing loss requiring cochlear implants. To bridge this gap in knowledge, this project goal was to develop a reproducible and translational in vivo double insult model involving acoustic overexposure followed by subsequent electrode insertion trauma that mimics the heterogeneity seen in humans. This model will be used to observe a functional and histological perspective to determine the harmful mechanisms at play and when, and eventually identify targets for future therapeutic intervention, and again, overall improving these CI outcomes. So to begin, I will review the translational and noise validation portion of this project. The first major part was developing in creating a translational noise exposure for rodents that would be similar to the noise you and I experience. And I emphasize this because in prior noise studies in our lab and in our field overall, the experimental noise protocols tended to involve exposing noise to singularly anesthetized animals with local ear inserts, so basically headphones. And this method proved limiting in several factors, including efficiency, translatability, and it added anesthesia as a disruptive independent variable given the confounding otoprotective capabilities. This generated our overarching goal for this part of the investigation, which was to characterize and validate a custom chamber with a rotating carousel in order to recapitulate noisy environments in awake and normally behaving rodents. The need criteria which shape the following design requirements are summarized here. First, we wanted to include a soundproof chamber big enough for multiple animal holding cells while still providing sufficient ventilation. The specific requirements for the animal holder system included a modular design that could fit multiple awake state animals unrestrained simultaneously with enough room to not induce additional stress to the animals, but also limit the movement to reduce potential for varied sound exposure. Additionally, we added a rotating component so that the noise level distribution might be ambient and uniform and more translational to the noise we experience on a daily basis. So this was the final product of what we were able to create based on this criteria. And once this design process was complete, the next step was to characterize the noise distribution throughout the entire chamber to ensure that the animals were receiving the intended intensity of noise we were administering. So our noise protocol we tested was a high intensity 100 decibel level noise, which it was a broadband noise, so 4 to 16 kilohertz for 1 hour. During this stage, we utilize a sound level meter, which basically is a fancy noise recording device. And we place this recording head in 4 different positions within one cell to mimic the potential or approximate positions of the rodent's head throughout the entire protocol. And in doing so, we are trying to estimate the noise intensity level that the rat would actually be experiencing on average. So during the stage, we established that each housing cell received an average dose of 112 decibels, so within 2 decibels of our intended range. The mean level of equivalence, or LEQ, which is the average sound energy measured over a given time period, only varied by about 2 decibels on average over the 3 trials for each cell. Using a multiple linear regression, we concluded that cell number, position, and trial did not significantly affect the LEQ measured during the 1-hour test period. Notably, but unsurprisingly, cell position had the largest effect among the 3 factors, but it wasn't statistically significant. I just wanted to present this finding because it's important to understand potential variability when doing the preclinical validation part of this project using the live animals, which I'll explain now. So this part of this study involved preclinical validation, which the overall objective was to establish that this new carousel design for awake noise delivery can be utilized to induce permanent hearing loss. To break this down really quickly, auditory brainstem responses were the functional test of interest and will be for all experiments going forward. ABRs measure the response to sound along the neural pathway, the peripheral auditory pathway. The information we collect for this data include thresholds of hearing, meaning what is the lowest intensity at which the ABR might elicit a response determined by the presence of the first wave or wave 1, which represents the peripheral portion of the cochlear nerve, so basically the nerve activity at the cochlea. Additionally, we were able to collect latency and amplitude responses of wave 1, which can provide sensory information regarding neuronal recruitment and conduction rates. After an auditory trauma occurs, thresholds, latency, and amplitude information can change from their pre-trauma baseline values. And by measuring this over time, we can make implications of how a trauma affects the cochlea from a functional standpoint. So having said all of that, ABRs were collected at baseline and at time points up to 28 days following noise, so 1 month. ABRs were collected across 6 different frequencies to capture any damage-related tonotopic effects. And these longitudinal differences in auditory function were compared to age-matched controls to examine proposed hypotheses. And we did only utilize males for this portion of the project. So here we have some of the ABR results showing the average threshold across 6 different frequencies, as I mentioned, at 3 different time points. So if we start for this left, we have our baseline value, baseline threshold values between the control in blue and noise in red. And then if we move to day 1 in the middle, you see that that noise group's average threshold shoots up significantly across all frequencies. And these thresholds remain significantly elevated at the day 28 time point. Although there is some recovery at all frequencies, especially in those lower 2 frequencies, 2 and 4 kilohertz, these values did not return to baseline or control values, and therefore a permanent threshold shift was established by our noise protocol. Also from this data set, we analyzed the latency and amplitude of wave 1, which I mentioned before, which is looking to assess for changes in cochlear nerve activity. Specifically, we looked at day 28 shifts, which represent the difference between the values at day 28 post-trauma and then their baseline trauma values. So we start on the left for amplitude shifts. The noise group was significantly affected. The shifts were negative because the amplitude at day 28 was lower than baseline, indicating a reduction in wave 1 amplitude that did not recover within the 1-month time period following noise. And this suggests a permanent loss of neural activity in response to sound. Similarly, wave 1 latency values showed incomplete recovery, particularly in these lower frequency regions. And unlike amplitude, latency shifts are positive, meaning the latency at day 28 was longer than baseline, so this reflects a delayed neural response to sound stimuli, likely caused by damage to the auditory nerve or synaptic connections. Overall, these findings indicate permanent changes in the central neurostructures responsible for processing sound. And this was expected based on the high intensity of noise exposure we administered and is consistent with similar findings in literature. The last thing I wanted to share for this portion of the project was some brief histology to further confirm our functional results. So we determined hair cell survival within the cochlea, and as expected, based on literature and the functional results I already presented to you, there was significant damage to the hair cells, specifically the outer hair cells of the noise-exposed group, especially in those middle and basal regions, and a similar trend in the apex, although this was not statistically significant. And then for the inner hair cells, they weren't as affected as the outer hair cells, and this was anticipated based on the noise protocol being used. This permanent irreversible damage to these morphological structures support our functional findings that indicated a permanent threshold shift. So to summarize the first part of this project, this data I've shown you thus far demonstrates that broadband noise exposure induces significant permanent threshold shifts across the entire frequency range we tested, with ABR thresholds remaining elevated from day 1 to day 28, confirming the model's ability to replicate long-term auditory deficits characteristic of noise-induced hearing loss. And now that we've established a sufficient noise protocol, we're able to design and execute the double insult model, which will involve administering an electrode insertion trauma via the round window of the cochlea as a secondary trauma to the neural substrate of the noise-exposed rats. This is for part A of the project, or this part of the investigation and the model establishment, and this really is going to focus on the functional standpoint. So,
is it always broadband, or do they use certain frequencies?
yeah, great question. If anyone didn't hear that, he was asking about the broadband noise exposure. So part of the reason we did pick broadband, so broadband means I think above a 4 kilohertz range, because that would be an octave band, but someone can correct me if I'm wrong. So we do 4 to 16 kilohertz because that's most representative of the noise we experience on a daily basis.
So if you're playing broadband, you play bass guitar, you're sitting on a bass. What frequency is that?
I don't know the specific frequency, but typically whichever frequency you're most exposed to is where you tend to see the most damage. So in some of those graphs, so I did a 4 to 16 kilohertz broadband noise. You see the most damage is between 8 and 16, and that's kind of characteristic of the noise that I administered. But for example, if you just did a notch noise protocol, meaning you just do it at one frequency, you typically see excessive hearing loss at that notch, so 8 kilohertz, or any like adjacent frequencies. A big part of this was just trying to sell to clinicians like, hey, we're doing something that's translational. So we mimicked central nerve hearing loss in rats that you see in humans.
You go to a Hurricane football game, everybody's trying to get the biggest speaker out inside the truck.
Yeah.
Really loud bass. Gone up to a couple of people and say, especially the children, say, don't you know that's probably affecting their long-term hearing?
And it is. Totally. Yeah, once you get up to a certain noise level, it doesn't matter what frequency is, it's just going to be damage. Okay. So this is the experimental design for the double insult model, which is exactly what it sounds like. It's a dual trauma consisting of taking a rat and collecting baseline functional measurements, which again are just ABRs for this project. The rats then received the acoustic trauma, same as the protocol I just showed you. However, the ABRs are then collected for actually 3 months following noise, at which time point they receive a unilateral electrode insertion trauma. So what that means is we're using an inactive electrode to do the CI surgery, so strictly mimicking the mechanical trauma of the CI surgery. And for our study, we aimed to only insert into the basal region of the cochlea. Again, following electrode insertion trauma, functional data was then collected for an additional 3 months, giving a total of 6 months, at which time point the animals are then euthanized and Cochlear harvested. This diagram kind of breaks down the two groups implemented in this experimental design, with the double insult group on top and then the EIT only control on the bottom, so no noise, but then EIT. And then for both groups, the contralateral non-implanted ears acted as the internal control. So for the double insult group, you would have a noise-only ear, and then for the EIT-only group, you'd have an H-match control. So 2 groups, 4 ears. And before I get into some of the longitudinal data I collected, I wanted to emphasize, again, the clinical relevance of my work and why this is really important for our overarching goal of therapeutic intervention. So this first graph is a representative cochlear implant candidacy audiogram, which I hope you remember from the beginning of my presentation. But as I mentioned, audiograms are the gold standard used by audiologists to evaluate hearing loss and determine candidacy. And one of the key aspects of my model is to demonstrate that the rats following the noise trauma demonstrate CI candidacy that mimics that of humans. So to do that with my data set, I've translated my preclinical data from my rat model into a clinically relevant audiogram format. And as you can see, at 1 month post-noise, the rats are already demonstrating this downward sloping ABR shift that persists, if not worsens, by 3 months. So their hearing is better in that lower frequency region 2-4, and then it drops off and stays down. So this just allows for a more direct comparison between the rodent data and the clinical audiograms used to determine CI candidacy in humans, and also demonstrates that we are in fact mimicking human CI candidacy. Now, to look at the entire timeline for the double insult model, the line graphs I'm showing are the ABR threshold changes for both the double insult ear, shown in red, and then the EIT only control ear in black, starting from baseline up until the 6-month terminal time point. And then the vertical line you see in the middle represents the time of electrode insertion trauma, so kind of splitting up the two traumas. And I also want to iterate that the y-axis is reversed for our traditional preclinical data sets, so that is why the line graph trend is reversed from my previous slide, and I'm happy to clarify for anybody. So this is a representative high frequency, 24 kilohertz, and you can see a significant elevation in thresholds in that noise exposed group immediately after exposure, as expected, and they remain elevated. And then at the time of electrode insertion trauma, you see both groups' thresholds shoot up and then remain elevated. And remember, we're inserting into the basal high frequency hearing region, so that's why we see damage in both groups, although there was some statistically significant differences in that prior noise exposed cochlea, which was interesting to see. Now, if we look at the lower frequencies, the noise exposed group again shows elevated thresholds prior to EIT because of noise, not as significantly as the higher frequencies, but that corresponds with the data I was showing you before. However, the EIT only group shows some sort of degree of recovery that the double insult group exhibits, whereas the double insult group doesn't exhibit this recovery at all. So based on these functional results, we theorize that EIT causes some sort of additive damage to the cochlear substrate, perhaps in part due to the compromised state of the substrate following noise trauma, which we believe is indicating that the apical regions are more susceptible to EIT damage when priorly noise exposed. And overall, these results demonstrate that the double insult model that we're trying to establish effectively mimics the prolonged and compounded hearing deficits observed in clinical cases involving prior noise exposure and cochlear implantation. Our initial immunohistochemistry observing hair cell survival unsurprisingly showed significant damage in all sections of the cochlea. We found significant differences between the full control group and the implanted groups as anticipated. And this is just very preliminary histology. So as we continue new and improved iterations of this model, we're hoping to find more links between the observed morphological differences and changes for hearing preservation CI outcomes, which I'll get into in just a moment. But to really just drive this model home, I want to show how these iterations have functionally proven that there are independent contributions of noise to functional hearing outcomes after EIT. So bear with me. So if we start on the left, we're looking at a comparison of the left implanted noise-exposed ear compared to the noise-exposed ear of the same animal. So same animal, but we're comparing the implanted ear versus the non-implanted ear, but they're both noise-exposed. So these were the ABR threshold averages at the 6-month terminal time point. And these findings indicate that the ABR thresholds are consistently lower, indicating better hearing, across all frequencies in the noise-only ear compared to the implanted noise-exposed ear. So this suggests that the electrode insertion trauma exacerbates functional hearing loss beyond what is caused by noise exposure alone, highlighting the compounding effects of the dual insult on the cochlear function. And if we move to the right, now we're comparing the left implanted EIT alone ear and the noise plus EIT ear. Both resulted in significant threshold shifts in the noise plus EIT in both ears in the high frequency regions. But if we look to the lower frequency regions, again, when we're comparing the EIT only ear to the noise exposed ear, there's some sort of spontaneous recovery that is observed in that apical region in those lower frequencies. So despite the electrode being inserted into only the basal region, functional deficits were observed in the apical low frequency regions. And this suggests that prior noise exposure may exacerbate cochlear damage or disrupt neural processing beyond the site of trauma, leading to widespread functional impairment. So this led us to part B of the project. Our functional results led us to design a round of experiments in which histological studies are to be conducted at various time points post-trauma to identify any sort of morphological changes associated with the unfavorable hearing preservation CI outcomes. So for this part of the project, we're implementing the same experimental design. So we are starting with baseline ABR measurements. Animals are randomized into the noise plus EIT or EIT only group. And then the group of animals receive noise, which leads us to our first time point in which these cochlea will be harvested 3 months after noise, so prior to EIT, and they will be used for H&E staining. And this time point was selected specifically to look at the state of the cochlea before implantation to solely observe how noise affected the substrate well after the trauma was induced, which may allow us to hypothesize or predict, based on the cochlear architecture, how might EIT further exaggerate these effects? And this is really translational and really unique to our kind of data set because when humans go in to get their cochlear implants, we don't really have a true understanding of what their cochlea looks like. So when they have this delayed acoustic hearing loss after implantation, we're not sure why. So that leads us to group 2, which is part of a set of acute experiments conducted at 48 hours post-EIT. And this time point was chosen to look at Western blots to observe changes in important inflammatory and fibrotic markers, specifically which are most active immediately following EIT. The next group of histology data will be collected at 1 month post-EIT, so around the time at which in clinic the CI would typically be activated for humans. And the idea here would be that we are able to observe the cochlea before electrical stimulation would start and affect the substrate even more so. So essentially when you get a CI as a human, you wait 1 month before activation. So you have a month to heal. So before you're activating, we want to see how is the cochlea healing after a month and how might noise affect that. And then lastly, the experiment termination time point, or 3 months post-EIT, 6 months. This is when in clinical papers there has been some observed initiation of the trend in delayed acoustic hearing loss post-implantation that I mentioned earlier. For all 4 groups, the contralateral unimplanted ears will act as controls for the noise-only and full control groups. And overall, we hypothesize that the cochlea in the double insult group will have reduced spiral ganglion density and decreased hair cell survival compared to the EIT-only ears, not only in those higher frequency hearing regions, but also in the lower frequency hearing regions. Additionally, we suspect the fibrotic and inflammatory markers will be higher in the dual trauma cochlea, and furthermore, we might observe how prior noise trauma to the cochlea might result in further exacerbation of these pathways, or rather, maybe how old scar tissue or loss of fibro ganglion may have developed over time, and how they may impact the functional outcomes we are observing. Once the previous histology I just explained is concluded, we really want to parse out how the neural substrate is changing after independent and then the multiple traumas over time. And then we aim to utilize a therapeutic regimen that can be rapidly translated from bench to bedside to counteract the negative consequences initiated at the time of surgical trauma. And with that, I would like to thank you all for attending this talk in the middle of your day or lunchtime, and I would be happy to take questions. Great question. So you asked about the timeline or the 3-month period. So one of the main reasons is because, when you and I experience noise, like you mentioned the football game, we go to the football game, the next day we're not getting a CI. So the idea is to make it as translational as possible. And this was long debated when we were designing these experiments, is like how appropriate is it to wait between the noise exposure and the CI. So 3 months was a trade-off based on aging and also because, again, we're not getting a cochlear implant a week after we get noise. So it was just kind of giving a long time period after the noise trauma occurred. I think it was between like 10 and 13 years for our brown Norway rats. Yeah. So we conduct experiments at 15 weeks. So, that's between 18 and 20 years for humans, sorry. And then that aging process is another 10 years. So still not directly translational. I'm not suggesting that when I was 18 and I went to a football game and by the time I'm 30 I need a cochlear implant, but that's kind of the trade-off with preclinical research, I suppose. 6 months is a long time period too, and the rats got to hold on for a while too. Yeah.
You mentioned in the translational noise validation that you only use male mice or rats.
Uh-huh.
Was there a reason or that was it?
There is one reason. Dr. Dietrich asked this during my proposal, but we typically tend to be partial to males because females have a protective effect of estrogen, which can negate some of the noise-induced hearing loss effects I was describing. And because this data set is nowhere else, no one's doing this, we wanted to limit any other confounding effects as much as possible. Convincing clinicians that this is translational is one of the biggest challenges of this project. So for now, females have not been used, but they need to be because it's still relevant. Yeah.
What therapeutics are you trying with? Variable data on
Sure. Great question. So there's a couple ways to answer that. So one of the biggest challenges with what they're seeing in clinic is, when you come in for a cochlear implant, you already have hearing loss. So, and as I mentioned, there's no FDA-approved therapeutic for hearing loss. So the idea behind the project is by understanding what's happening at the time of the surgery, we'll be able to create a therapeutic regimen surrounding that, and we're still figuring that out. Now, another part of this is our lab is known for or has published literature in implementing mild therapeutic hypothermia as a therapeutic to both negate or mitigate noise-induced hearing loss effects as well as cochlear implantation post-surgery delayed hearing loss. Sure. Yeah, they can. I think there's a couple different regimens. I won't speak to them specifically, but I know in some cases they'll administer like a corticosteroid to sort of counteract any inflammation. Obviously, there's pain meds involved, but it's not helping. and that's the whole point of this project, is understand what we need to do differently. So is the time point at which we're administering a therapeutic what's important? Is it what therapeutic? Is it a combination of therapeutics? So, I've described both the pathophysiology of electrode insertion trauma and noise-induced hearing loss. There's a lot of overlap. So I think it really will depend on the time point. Is it at the time of surgery? Is it during surgery? Or is it before? Or maybe it's all. So that's why those time point-based experiments, I think, will really help us understand what's going on over time following the respective traumas. Yeah.
To make your model even more translational, have you thought about this, so we're talking about the fact that it's not just one exposure that we need to have a cochlear. Have you thought about multiple exposures to noise, pre-SCI?
Definitely. So the question was about the experimental design regarding noise exposure. So we do a one-time noise exposure. Obviously, that is a challenge in terms of translatability. We don't just go to one football game our whole life or one concert our whole life. But what I was really trying to mimic here, which I was showing earlier, which I was comparing the human CI audiogram to my rat audiogram, if you will, was just to show we're mimicking the hearing loss profile, not necessarily the constant noise exposure. And again, I think this is just sort of a preclinical limitation. However, we are discussing the potential for future iterations of this model, specifically in other noise-induced hearing loss studies we do, is doing a repeated exposure and seeing how that changes the hearing loss profile, how that changes the morphology of the hair cell loss or the spiral ganglions, and something similar to that. But that's a really good question, and that would be a great next step for really building out this model.
Went to a movie the other day, Bob Dylan movie, you seen it?
I don't think so.
Very good. My point is that I'm sitting over the speakers, and the Dylan movie didn't really affect my hearing, but they have some children's comics in the early and the street noise. I could feel my ears just floating. So when I got home, I flew my ears. So
That's great. Definitely. Thank you, guys. Thank you so much.